Okay, so PCR is the next biotechnology we're going to talk about briefly. We'll be talking about this in lab and actually performing it in lab and doing some forensics types calculations. So I'm going to go over this briefly. Kary Mullis invented PCR in 1983. He was doing research for a biotech corporation and he just came up with this idea. I think he was doing sequencing and he came up with this idea of what if I had two primers to target a certain site, then I could exponentially synthesize more copies of that targeted location. And he invented PCR. He won the Nobel Prize in 1993, and he was a quirky guy. I heard that he sold the rights to PCR for something around $10,000 to $15,000, which was a mistake, but at the time probably seemed like a lot of money, and he didn't know. Maybe he didn't know what he had. But what PCR is, it's an in vitro DNA synthesis amplification with exponential synthesis of the target DNA sequence. So let me say that again, an in vitro DNA synthesis that is targeted with amplification. That means that you make millions of copies, but just of one targeted DNA section, not the entire genome. So it is based on the concept of what we know about how DNA replication happens in the cell, but then by adding synthesized primers, we can cause the primer to stick to a very specific location on a chromosome. And there's actually two primers that create like bookends. And then the synthesis goes just between those two primer sites. You do have to know the sequences, at least of your primer sites, before you can do PCR. So there has to be some sequence knowledge already there. But there were ways to figure those things out, and we've improved on those things as time has gone on. But like the discovery of restriction enzymes, PCR was really a huge game changer for all of biotech. So even if you started with just one copy of a template, and the template's usually going to be whole genome, the entire genome of a cell or a couple of cells that maybe you swabbed off of a blood spatter at a crime scene or a cheek cell swab you took from somebody, then you can extract that DNA. There's now very quick ways to extract DNA from cells. And then what you do is you take that DNA, you mix it with these pre-synthesized primers in a test tube, and then you also add nucleotides and you add an enzyme that is heat resistant. So there's a couple of naturally occurring polymerase enzymes that are heat resistant. And so you put these test tubes into a machine that heats and cools the test tubes. The first thing that's going to happen is the DNA is heated to about 95 degrees. That's going to separate the two strands of the double helix. And then there's a cooling where the primer is able to get in and base pair with its complementary site. And then the test tube is heated a little bit again, and that allows the polymerase to reach its optimum temperature and it synthesizes the new strand. And that's considered one cycle. So this is one cycle. After one cycle, you have twice as much template as you had initially. And then each additional cycle, you boil the DNA again, it separates the two strands, and each of them can be used as a template again. So with each cycle of heating and cooling, you're doubling the amount, but just of that target sequence that you have. And then ultimately what you're doing with that, sometimes you're cloning that into a vector. But for human identification, what we tend to do is we use primers that are on either side of repeat sequences because they're highly variable in the human genome. And so by using primers that lie on either side of known repeat sequences, we can measure the length of those sequences by doing the PCR and then running the PCR products on an electrophoresis gel. And what that does is it gives us a pattern for one locus for a certain individual. And we repeat this. The CODIS system uses 13 loci and it tests individual. And in this example, each of these individuals is homozygous for their repeat sequences, but really in real life, individuals can also be heterozygous. And so you get these patterns of different PCR fragment sizes at different locations that collectively creates what we call a DNA fingerprint for one individual. And so in lab, we'll do some calculations to show that your DNA fingerprint is unique to you beyond any doubt mathematically. All right. So DNA sequencing, this is a technique that was invented pretty early on. The first version of this is called the Sanger method or dideoxy sequencing. And it's any kind of sequencing technology is a chemical reaction that allows scientists ultimately to determine the nucleotide sequence of any particular DNA molecule. So it could be one of those clones that you have created in your library and put into the freezer. You take one of those clones out, you extract the DNA, and then you run it through the sequencing method. And really, the first method, the Sanger method, required the use of radioactive nucleotides, followed by gel electrophoresis, and then the exposure of that radioactive gel to a piece of X-ray film. So the first couple genes that I cloned were done this way. It's radioactivity is somewhat hazardous, and there's a lot of training for the handling of all of that we had to go through. The film was then developed after, I'd say, 24 hours-ish, and we just use a regular X-ray developing machine that you might have, for example, in a hospital. So some of you might be familiar with that. And it gives you a result that looks like this. So this is an old-fashioned kind of DNA sequencing gel radiograph. So it's an X-ray film. And what you can see is the sequence of DNA, but you have to read it from the bottom up, and each lane on the gel only represents one nucleotide type. So I'm not going to go through the explanation of how the chemistry works, but ultimately when you read from the bottom up, you look at what lane you're in. Cytosine, thymine, adenine, and guanine is the way they loaded this gel. The DNA sample sequence from a healthy person is here on these 4 lanes on the left. The DNA sequence from somebody with cystic fibrosis is on the right, and there's a small deletion that you could find if you sequence from the bottom up and you compare the sequences to each other. And so this is how we started doing these kinds of things, but not in the late 80s, early 90s, they developed an improved version of this. Instead of using radioisotopes, they used fluorescent molecules for the nucleotides. And so this was much safer, and you could load all of the samples onto the same electrophoresis lane because there was a laser that would read the colors, and so you could tell which base it was. And so it would read from the bottom up, just like the old-fashioned type, but it would you could do a whole sample in each lane, a whole different sequence of different fragments. So you could get a bunch more onto one gel. This is how we did the Human Genome Project. So this was the technology that we had at that time. At that time, this cost quite a bit of money. A single machine doing this kind of sequencing method was $100,000. And we had a very small lab at UT Southwestern. We had about, 15 or 20 machines. Some of the larger labs at some of the universities in other places in the US and around the world had, 40 or 50 machines. So there was a lot of investment in the equipment. Ultimately, that technology was phased out by what we call next-gen sequencing, which came out around the end of the Human Genome Project. They realized there was going to have to be improvements to the sequencing because we got stuck on certain parts of the human genome that we just couldn't sequence across. We couldn't get sequence for certain sections. Basically, a lot of them were repeats. Really long repeat sequences that the Sanger sequencing just couldn't manage. And so they developed some next-gen sequencing, and one of the next-gen, the first one that was invented is called Illumina. That was invented right about 2001, and that's what's shown here. It's fundamentally a different kind of sequencing in a lot of ways. There's no electrophoresis. You attach the fragments to the wells of a plate, and then you attach little adapters to beads, and then the DNA fragments are attached to those, and then you wash different nucleotides across the top, and there are different signals that come out of the wells that are read by a laser. And ultimately you get a computer reading the sequence of 96 different target fragments at once. And so it was faster, it's more accurate in some ways, and you can get longer sequences as they improve some of this technology. You can get longer sequences. So this is how this Illumina sequencing works. In this picture, they're showing you like you're looking from the top down into one of these plates. In this example, there's only 9 wells in this plate, for example. And when they wash the nucleotides over top, this well over here, let me get my pen. This well here incorporates an A, this one incorporates a G, this one incorporates an A, and it's letting out a little light, a little beep of light, and then red means A, green means G, yellow means C, and there's a computer keeping track of which well is emitting which light. And so and then in the second cycle, they wash the nucleotides over again, and you get a different nucleotide being added to each well. And ultimately, the computer is keeping track. So in well 1, it ends up being AGT. You can see in well 1, it is A in cycle 1, G in cycle 2, T in cycle 3. So the computer is keeping track of all these little blinks of light that are happening, and you're getting sequence of typically 96 different fragments of DNA at once. This is a typical plate that we use in molecular genetics. It's 12 wells across, little dips. It's 8 down, and so it's an 8 by 12 grid, and so that's where I get 96 wells. And so this one is more of like a PCR type plate. You could do 96 PCR reactions. This one could be used for other things. It doesn't have such deep wells as the other one, but you use these for different things. They're made out of plastic, and you actually have pipette men that can do 8 channels at a time, and you even have machines that can do 96 channels at a time. You put a whole box of tips on, you suck up whatever liquid you want, and then you put your plate in and you deliver that liquid into the plate. So there's a lot of shortcuts. We don't have to do everything manually with a single pipette man. But as you can imagine, you can ramp up any of these technologies and get a lot of reactions happening in a pretty short period of time. And so that's really what happened with the Human Genome Project. You started to be able to, as the technologies improved, you started to be able to get this enormous amount of data. And then what was not there was the ability, the computer programming ability to make meaning of the data. And so the birth of the bioinformatics movement with big data happened about the same time as these genome projects and these improvements in sequencing, because now we're starting to be able to get huge amounts of data in very short periods of time, but we didn't really know how to get the computer to make meaning out of it. So that was the beginning of what we call bioinformatics, which is really considered to be a field of computer science. But we also have computational biology, which is really basically biologists learning enough of the programming to try to do some of their analysis on their own. So either one, if you're more of a computer scientist, but you know something about the biology, you could go with a bioinformatics route. If you know a lot of biology, but you're interested in programming too, you can be a computational biologist. So 2 kind of different emphasis on similar types of things. All right, so I'm going to stop this part of the lecture here, and we'll pick up with some of the mouse techniques in our next segment.